Good afternoon and hello everyone. My name is Fyp1 and in this video, I'm going to present on my final year project entitled Microbial Profiling of Dropsy Disease in Purao. First, I want to drop a quick appreciation to my supervisor, Dr. Chu, for the help throughout my writing on the proposal and the guide for this presentation as well. So back to the presentation. This presentation will consist of introduction, literature review, materials and method, expected outcomes, and the work schedule that I have prepared for this study, together with the references used to complete these slides. So, in this first part of the introduction, why do this topic should be a concern? It is because infectious abdominal dropsy is known as one of the aquaculture disease. From several studies, aquaculture sectors have followed an increasing trend of development worldwide, including our own country, for food security and demand of the human population on fisheries product. The occurrence of dropsy will have high tendency to hinder the development of aquaculture and cause economical losses if not prevented. It will also reduce the potential of Ampurao fish for market and propagation in the aquaculture industry. Therefore, the objective of this study is to perform microbial profiling assays on dropsy disease Ampurao and to identify the actual causative agents of dropsy in Ampurao fish. From that, it is hypothesized that the dropsy disease Ampurao will have the same causative pathogens of dropsy in other fish. The next hypothesis is that Aeromonas hydrophila and Pseudomonas fluorescens will be found in the microbial profile of the dropsy disease Ampurao. Next part is on the literature review, which will touch on the Ampurao fish, dropsy disease, the possible causative agents of dropsy, and what is microbial profiling. So, Ampurao, or its scientific name Thothambrodus, is locally known as Kelah, and its English name is Malaysian Masir. This fish is used in this study because they are still new to aquaculture sector and have significant importance, especially in Sarawak, for food, ornamental, and sport fish. This fish used to cost about 400 ringgit per kilogram, but recently it is said that the price range can reach between 800 to 1000 ringgit per kilogram. This fish can be distinguished by looking at its silvery bronze body color with dark reddish fin. It also have long lower median lobe and pointed rostral hood, which differ them with other tor species. Now, I will move on to dropsy disease. This disease can be identified by looking at the stated symptoms, which is swollen belly. For your information, this is the main signs of dropsy. The other symptoms are hemorrhagic lesion, scale loss, imbalanced movement, and exothalmic condition, which can be seen in the picture from this slide. The mortality rate of this disease is high with strong spreading potential, which is why it is common in aquaculture fishes. The causes of dropsy other than microbial infections is it can be caused by fish low immune system and rapid fluctuation of environmental factors, as this can lead to invasion and growth of the causative pathogens in the aquaculture environment. Next, on the possible causative agents, during the 20th century or 1980s, some researchers assumed that dropsy is caused by bacterial infections. Some said it was viral infections, and the rest believed that it was a product of both bacterial and viral infections. However, recent studies shows that Aeromonas hydrophila and Pseudomonas fluorescens are the most common causative agents found in dropsy disease fishes. Now, for the last part of the literature review will be on microbial profiling. Microbial profiling in this study will involve several methods that will allow characterization of the microbes morphologically, biochemically, and their molecular identification. The morphological methods will have gram staining and microscopy assays, while the biochemical methods will be using API-20 and EKIT. As for the molecular method, 60S rDNA sequencing will be utilized in this study. Next part is on the materials and methods that will be applied in this research. This slide will show the overview of the methodology. For sampling and sample preparation part, the fish sample will be provided and taken from Dr. Chung's lab. The fish muscle tissue sample will be cut by using sterile knife. The cut sample will be crushed using a sterile mortar and diluted to be used throughout the experiment. Subsequently, for the isolation of the bacteria, the procedure involves streaking of sample on agar plate. The agar type to be used are Aeromonas isolation agar, RS agar, and McConkey's agar. Next is on the gram staining. This procedure is done to classify the bacteria from the sample into positive gram and negative gram bacteria. This will allow us to know the thickness of the bacterial cell wall, which is part of their morphology. Gram-positive having a thicker cell wall while gram-negative have thin cell wall. The procedure and several reagents will be used in this assay can be viewed in the following pictures. As for string test, this test will be used to identify gram-negative bacteria, whereby gram-negative bacteria will give a positive result, causing the visibility of the potassium hydroxide string. On the other hand, positive gram bacteria will give out a negative result in which the potassium hydroxide string is non-visible. The positive control that will be used in this procedure is using E. coli, while negative control will be using Bacillus spp. For comparison of bacterial growth part, this procedure is crucial to identify the favorable environment for the bacteria. This is because different bacterial strains will have different optimum growth parameters value. The optimal value for bacterial growth using each parameters can be obtained by observing on what value the colony will grow the biggest. The parameters involved are temperature, pH, and salt concentration. For biochemical characterization, using API-20 and kit, this kit is used to identify non-fastidious, non-enteric gram-negative rods through a biochemically standardized system. The example of bacteria that can be identified using these kits are Pseudomonas, Acinetobacter, Flavobacterium, Vibrio, and Aeromonas. The main components of the kit is a strip which have 20 miniature biochemical tests. This is where all the biochemical tests will be conducted. Next is on the DNA extraction, which we will be using phenylchloroform assay. The principle of this assay is liquid-liquid extraction of biomolecules. It is function to denature protein portion of a cell and removes it, separating genomic DNA into a soluble phase. Furthermore, it helps purify DNA to further carry out the bacterial characterization procedure. The procedure is as shown in the slide. I will now proceed with amplification of 16S rDNA using PCR. This procedure will selectively amplify DNA targets using polymerase chain reaction (PCR). The procedure consists of repeated cycles of heat, denaturation, primer annealing, and primer extension. This assay have complex hybridization which helps identify bacteria at the species level, and it also assists the differentiation between closely related bacterial species. Now, for 16S rDNA sequencing, this assay is used to sequence the 16S rRNA gene that codes for the small subunit of the ribosome found in the bacteria. These assays will be conducted by a third-party service provider. The flow of this procedure is as shown in this picture. The last part on the methodology, which is on the 16S rDNA sequence analysis, will be using BLAST tool provided by the National Center for Biotechnology Information (NCBI). The tool can be accessed by using the provided link in the slide. This tool works by finding regions of similarity between sequences, then compares it with 16S rDNA sequences from bacteria in the databases. The phylogenetic tree for the bacteria can be obtained by using MEGA 4.0 software. Next, the expected outcome from this study is to successfully isolate, extract, and profile the causative agents of dropsy in Empurau fish. Moreover, it is in my expectation that Aeromonas hydrophila and Pseudomonas fluorescens is the causative agents for this disease in Empurau. Furthermore, I also expect this study can provide beneficial microbial profile for further research used by the Fisheries Institute and aquaculture researchers. Lastly, from this project, the right precautionary measures or steps to avoid occurrence of dropsy in aquaculture fishes able to be provided to improve their economical value and quality in the future studies. Now, here is the work schedule that I have planned for this particular project, which starts from October 2021 to July 2022. This work schedule have also been reviewed by my supervisor and will tell the details on the flow of my final year project. And lastly, these are the references that I used to prepare these presentation slides. I think that is all from me. Thank you for watching and have a nice day.